So this online lecture is hitting a couple topics in Chapter 4 that sometimes seem challenging for students. I am not going through all the organelles. That is up to you to do. You have an organelle chart. I'll remind you of what that looks like in a minute to fill in, and you can get that information from the textbook and or the videos that I have provided for you on the Chapter 4 summary page. So the first thing I want to talk about is cell theory. And I always find it challenging when I'm learning some history of science to remember that there was a time when we didn't know all we know now. It's hard to imagine not knowing about cells. But before the 1800s and the invention of microscopes, scientists, people didn't know that we were composed of these little organized units called cells. And once scientists were able to start seeing some of these cells, they came up with the cell theory. And it's really simple, and most of us go, duh, but I want you to appreciate that this was a huge breakthrough. Like I said, in the mid-late 1800s. So the first part of the cell theory is that all living organisms are composed of one or more cells. So yes, there are unicellular or single-celled organisms. A lot of these are aquatic, like amoebas, parameciums, euglenas. I'll show you a couple pictures in a minute. And there are multicellular organisms, like you and me, and plants and animals and fungi. Things that are made up of more than one cell. Okay, so that's the first part of the cell theory. The second part is that cells are the smallest unit of life. So back in Chapter 1, we talked about some of the characteristics of life, like using energy and grow and develop and respond to the environment. And you were challenged, potentially, depending on which questions you got in the Chapter 1-2 quiz, about deciding what a certain object had that was or was not characteristics of life. And I asked you in homework to go through the characteristics of life and decide if viruses showed these characteristics or not, and which they showed and which they didn't. Because there's a lot of non-living things that, for instance, use energy, right? Our cell phones. Things that respond to environment. Our cell phone, when you drop it in the bathtub, yeah, it dies. But they're not made of cells, even though it's called a cell phone, right? It's not made of this organized little membrane-bound unit. That is why my argument is that viruses are not alive, because I take this literally as the definition of life is that you have to start with a cell. But as you went through that exercise, you probably learned that viruses do show lots of characteristics of life. And so some scientists consider viruses living. The third and most important, and was the hardest for people to grasp, part of the cell theory is that new cells only come from pre-existing cells. Okay, so new cells come only from pre-existing cells. It means you have to have started with a cell to get more cells. And they get there by cell division, which we will talk about later this semester. We'll talk about, binary fission and mitosis and meiosis. This helped change people's minds about the idea of spontaneous generation. And the classic example is when maggots appear on meat that you have left out on the counter, right? So they did experiments like, where did these maggots come from? Spontaneous generation. They just came out of the air. And we now know that's not true. And what was really interesting for me is this past summer, we were at the Pacific Ocean and we found this dead seal. And, it was kind of gross, but kind of cool. And we looked around the body and we saw all these maggots, all these white little things just wriggling around. And my daughter was like, where'd they come from? And my first response was like, wow, I don't know. And then I had to think, okay, you are a biologist. You know that flies, some kind of bug, had to lay these eggs, and this is where all the maggots come from. But sometimes when you're looking at things, you kind of forget these rules of science. And it was like, wow, they just appeared. But that's not true. So the cell theory, again, is kind of one of those, well, duh, we know this. But remember, this was really groundbreaking, that cells were the smallest unit of life. All living organisms are composed of one or more cells, and new cells come only from pre-existing cells by cell division. And I want you to remember that cells are really small. So if you take a look at your hand or your arm, you see lots of features, but you're actually not seeing a single cell. So to see most cells, we need a microscope of some kind. And yes, there are some examples of big cells, like eggs, but those are really the exceptions. Okay. Bacteria, which we'll talk about, are prokaryotic cells. Those are even smaller. And then to be able to start seeing inside the cell, the organelles, to see viruses, which are teeny, you had to get even more complex, more complex types of microscopes. So let's talk a little bit about microscopes. And before we go there, I want to remind you that cells are not all circle, square, oval kind of things that we draw, right? Cells come in lots of different shapes and sizes. So this is a skin cell, and you can see it's kind of stretched out, and all these fibers are the cytoskeleton that you'll learn about. This image in the black and white is multiple neurons, and here's the cell body, and there's all these projections. Okay. This guy is an amoeba. Amoeba. I always spell it wrong. Isn't that sad? I always spell it wrong. All right. I know there's an O in there. And look, he's got this really cool shape because he crawls around. He is he's not a he, but it is a single-celled organism. Just like this little guy here, he's my favorite. This is a euglena. This is a single-celled animal cell that can actually do photosynthesis. Really cool guy. Love to use it as a test question because it confuses people. No, it doesn't confuse you, I should say. It makes you think. These are plant cells. The little green in here are chloroplasts. And this guy is a macrophage. Let's see if I can write. Macrophage. That's going to digest some bacteria. Macrophages are part of your immune system, and they move around your body looking for foreign invaders to eat up. So even though we're going to draw our animal cells and our plant cells and our bacteria like these funky little shapes, remember that they are very diverse in their shapes. And what do we always say when we use the word shape? Shape or structure equals function. All right, so the two general types of microscopy I want you to understand is this guy that you have used, I'm sure, before in some type of biology lab, the light microscope. It's also called bright field. Okay. Fairly simple microscope. You have light coming through your sample. You have objectives and eyepieces that magnify the sample so it looks bigger. The resolution, we'll talk about what this word means in a minute, for a light microscope is about 0.2 micrometers. Over here, this fancy guy is an electron microscope. And, like the name implies, microscope, instead of using light to let you see your image, your cells, it uses electrons. Okay. It has a resolution of about 2 nanometers. So you have to remember that 0.2 micrometers is the same as 200 nanometers if you do your conversion. So an electron microscope can get about 100 times more resolution, more details than a light or bright field microscope. A disadvantage of an electron microscope is that you have to kill your sample. So the cells have to be fixed in order to shoot the electrons and get the image. Whereas a light microscope, you can look at live cells or you can look at dead and stained cells. Now let's talk about cells a little bit. So with light microscopy, you can see cells, and I shouldn't say just no smaller, but a few organelles. Okay. So for instance, this that I'm going to outline in red, this is a cheek cell. And these little purple squigglies are bacteria. And I hope you get to do this in lab. I think it's totally cool when you scrape some of your own cheek cells, get a little of that bacteria off your teeth, look at it under the microscope. So one thing I want you to notice is with a light microscope you can see bacterial cells. These are bacteria over here. But you definitely cannot see any details of the bacteria, right? Because they're so small. The other thing you can see with this light microscope is this little guy is the nucleus. So that's why I say you can see a few organelles. Okay? Over here in a plant cell, here's a single plant cell. These green little guys in here are the chloroplasts for photosynthesis. So that's an organelle you can see. And this little guy over here is another single-celled organism called a paramecium. And yes, in fact, there are some organelles that you can see in here. You can't distinguish them very well. You can, if you look closely, see some little hairs. That's the cilia we'll learn about that helps this little guy swim. But with light microscopy, you cannot see a lot of detail. Okay, it just doesn't have the magnification capabilities. So if we want to see detail, we'll use an electron microscope. Oops, and I lost one of my words. So there's two types. There's a TEM, which is transmission. I don't know why it does that. Transmission electron microscopy. And there's SEM, which is scanning electron microscopy. And what I want you to see is the difference in the details. So here now, this is the outside of our cell. Here is the nucleus. This dark spot in the middle is the nucleolus, which you'll learn is a region in the nucleus. And then you can start to see these organelles. And you can actually start to identify them like they are here. There's a vacuole and a mitochondria. And you can get a lot of definition with electron microscopy. So transmission gives you what I call a 2D image, right? It's kind of a flat surface. You're looking right through your sample. Scanning gives you a 3D image. So here are some red blood cells, and here's some, I think this might be a clot, so these might be fibrous parts of the clot. This is some kind of immune system cell, maybe a macrophage. So you can see this 3D, you can see the shape, the surface from scanning. This, whoops, is a chloroplast. And as you learn about chloroplasts, you're going to learn that they have all these internal membranes. This is where our photosynthesis is happening inside this organelle. So lots of detail that you can see with electron microscopy that you can't see with light microscopy. Let me make another point here. Okay, chloroplasts, naturally green. This is natural color. Same with the color you see here, natural color. Okay. These cells are stained. So your cells and bacterial cells are basically see-through. So they're really hard to look at under a microscope unless you do some kind of staining. And once you stain cells, then they're dead. Okay? So when you look at paramecium or chloroplast in a leaf cell under the microscope, you can actually see movement. And it's really cool. I hope you guys get to do this too, where if you shine enough light on the chloroplast, they start this cytoplasmic streaming, and the chloroplasts start kind of moving around, trying to get the best amount of light. Electron microscopy is black and white. That's what your images look like. You can't stain them. So if you see something like this, which is really pretty, this is called false coloring, which is basically like a coloring book, whoops, that they add colors to certain regions after they've taken the image. Okay, so you've got two types. You've got light or bright field, you've got electron. Magnification is super important when reporting images. So cells at different magnifications. Here's a plant cell using 100x total magnification. Here it is at 400x. Right? You get a lot more detail. And then, this wasn't the best example, but here is a plant cell under the electron microscope where you see lots of details. So for instance, you can't see the nucleus, right? It's not stained. It's just see-through like everything else. Here, because of the way the electrons take the image, you can see the nucleus and the nucleolus. And you can see more defining things like vacuoles. Most of the time, and these again are bad examples, most of the time there's a scale bar that'll tell you this is 1 micrometer or 1 nanometer or 1 whatever, not nanometer, that's silly, whatever size. You always want to report your magnifications, right? So if you're looking at stuff in the lab, make sure you remember the magnification, which with a light microscope is the ocular, which is usually 10x, times the objective, which you will probably be using either a 10, a 40, what's in between? Maybe 5, 10, 40, 100. So that your total magnification, 100x, would be from a 10x ocular and the 10x objective, 10 times 10. And your 400, would be the 10x objective. The 10x ocular, the eyepiece, and the 40x objective to get a magnification of 400. So make sure you understand how to calculate magnification given some information. Another term that goes with microscopy that I want you to understand is resolution. Resolution is the ability to distinguish two objects that are close. The ability to distinguish two objects that are close by each other. Okay. So here the objects are what we call resolved. Here you can't tell that they're distinctly two different objects. So again, the resolution limit on a light microscope is about 200 nanometers. So that means if they're closer than 200 nanometers, you can't tell them apart. And with an electron microscope, is 2 nanometers. So 100 times more powerful at distinguishing different parts of a cell. The other term that we use with microscopy is contrast, right? So if you look back here, I can't see anything on my plant cell except for the chloroplast because they have big contrast color differences between the cytosol and the other organelles in the chloroplast. Likewise here, to get the contrast, we use staining. There are some microscopes that have what we call phase contrast. So these are light microscopes, and they allow for shadows to be seen. So the nice thing about phase contrast is you can start to see more detail without having to kill the cell. Okay, when you stain it, you have to kill it to add the dyes to it. So contrast just in general means, can you pick things out differently? Can you see the details, right? Because here with just light microscopy, hard to see. I mean, here's the nucleus, but it definitely stands out more with phase contrast. So just some terms about microscopy that I want you to be familiar with. Let's talk in general about the cell types you're going to be looking at. So we have prokaryotes and eukaryotes. Prokaryotes in general are much smaller. So again, we think of bacteria, but there are some really cool ones called archaea that are the ancient bacteria. But these little yellow guys, and this is a scanning electron microscope image with false coloring, so the bacteria aren't really yellow. Okay. And these are again stained, so the big eukaryotic cell and the small prokaryotic cell. Typical bacterial cell, no nucleus, no membrane-bound organelles. Okay. Okay, we'll talk about that in just a second. But they do have a cell membrane, also called the plasma membrane, and they do have DNA, and they do have ribosomes and cytoplasm. And even though these cells, people say, are much simpler, what I want you to remember is that a bacterial cell can do everything your eukaryotic cells can do. So bacterial cells actually do cellular respiration and all that kind of metabolism and protein synthesis. They just do it along this one plasma membrane instead of having all these organelles like eukaryotic cells. So eukaryotic cells, we typically talk about plant and animal, but the fungi, those single-celled protists, those are all eukaryotic cells. And the big difference is eukaryote means true nut. And the nut stands for nucleus. Prokaryote means before the nut, before the nucleus. So your eukaryotic cells are always going to have this nucleus where the DNA is located. They also have cell membranes and cytosol and ribosomes and DNA, but then they have these other membrane-bound organelles. So they split their functions into distinct organelles, where prokaryotes do it all in one cell. One space. They're both one cells. All right. So what I want you to do for your eukaryotic cell, and there's actually 2 pages to this. I gave it to you as a handout. It's also under this week's module. I want you to fill out this chart. So, plasma membrane. I just told you, you find it in all types of cells. And then you got to figure out its function. So I'm going to do this first one for you. So the function is to contain, actually let's write this different, to separate the environment from the inside of the cell. Right? That's the function. That's what its purpose is. So I want to make sure you guys are understanding that. You could also put the function is to control. This isn't this. Okay, so I just can't write little. What goes in and out of the cell. Right? And the description, I want you to describe something about this. So my description of a cell membrane is it's a phospholipid bilayer. That means it has a double layer of phospholipids that does this function of separating the environment from the inside of the cell, controlling what goes in and out. So please, please, please work on this chart. All right, 3 more concepts I want to go over. Why are cells small? Cells are small because of diffusion. And diffusion is just the movement of molecules from high to low concentration. Movement of molecules from high to low concentration. So this is random, and the coolest way to look at this is if you take a glass of water and you drop some food coloring in it. Eventually the food coloring will change all of the water that specific color. That's diffusion. It's just this random movement of molecules, and they like to get away from the high concentration. They go towards the low until it's all at equilibrium, where the concentration is equal. So if you have a large cell, and say you need to get some sugar in, and it goes diffusing in, now I'm inside the cell, and it's going to take a long time to get where it needs to be. Whereas a smaller cell, that slow diffusion, same rate, can get where it needs to be a lot faster. So one of the things we talk about is the surface area to volume ratio. And some people write it as ARG. Surface area to volume. Okay. You want a large surface area compared to your volume, because that way lots of things, I don't know what that was, can get in where they're needed. So here's a little example of calculations, right? Surface area to volume, and as you increase the volume, right, you don't necessarily increase the surface area as fast. Right? So you want a large surface area to a small volume. Get things in and out quickly. Another super important concept I want you to understand is the difference between genome and proteome. So I want you to remember that genome is your genetics, your DNA. And when you start out as a little egg and a sperm and become this diploid cell that divides and divides and divides into you, the DNA in all your cells is the same. Now, we're not talking about mutations happening, okay? But I want you to understand that your muscle cell, your nerve cell, your blood cell, your skin cell, they all contain the information for eye color and hair color and curly hair or straight hair. But they don't show that, right? Your muscles are not hairy and blue if you had blue eyes because of the proteome. This is the proteins that are expressed. So just because you have all this information in each of those cells doesn't mean all of it's going to be used. So I want you to think about your genome like a cookbook. Okay? So say you get this cookbook with all these different recipes. You're not going to make everything in that cookbook for dinner, right? You're going to pick a couple meals. Maybe you're going to make some lasagna, or tomorrow night you're going to make some tacos, and the next night you're going to make some goulash, and next night you're going to make some pasta, right? Each meal is like a different cell type. You're only reading part of that cookbook. You're only expressing some of the recipes. So you still have that whole genome in there. It's still there. But to make lasagna for dinner and tacos and goulash the next night, you're only going to use certain bits of the information. So all your cells have the same genome, but all your cells are unique because of their proteome. They're different proteins expressed. It's a super important concept, so just kind of let that sink in a little bit. The final topic, and it's kind of interesting to me that it's here in this chapter. We'll talk more about it. Where are we talking about it? Did we talk more about it? I'm looking at our schedule. We'll talk more about it, in chapter 10, gene expression. But this figure, that's figure 430 if you have the textbook, is about protein synthesis. And one of the objectives for this chapter is to understand that proteins. Wait, I'm trying to look for the... Dun, dun, dun. Hold on. Oh, maybe it's not on our objectives. Series of components. Well, that's kind of awkward. Let's just look at this and say, what a beautiful picture. This is a huge part of the cell biology class that I teach. And the important thing I want you to understand, let's just leave it at this for right now, is that when you learn the function of ribosomes, you're going to learn that on the rough ER, it is rough because it has ribosomes. And you're going to learn that the function of ribosomes is to make proteins. It's called translation. And I want you to know that translation can happen in the cytosol or on the rough ER, depending on where that protein is going to go in the cell. Okay. So proteins that are destined for the nucleus, mitochondria, chloroplast, peroxisomes, or that function in the cytosol are translated on ribosomes that are free floating around. So if this was my cell, my nucleus, and I had all these dots, these would be the ribosomes. There's also the rough ER, which is this series of membranes that have ribosomes on them. So there are two sites of protein synthesis. Synthesis just means making proteins. They can be free ribosomes or they can be ribosomes on the rough ER. All right, so go learn your organelles. We're going to have a fun activity to help you remember all these guys, but it's super important to be able to recognize structure, understand function, and know what kind of cells these are found in, because we're going to build the rest of the semester on the functions of these different organelles. All right, that's it.